All right, good morning, everybody. Happy Wet Monday to you. All right. So we've got a bit to do today. Here's the short term. Today we talk about what we talk about today. Next class we talk about bioinformatics, different chapter. Friday we've got a review session, and then Monday is our first test. Okay, so that's where we're headed. So we've got today, and next class will be the content that will be covered up till today, and the next class is all the content that will be covered on the first test. Some of you are maybe still working on this, but these were the three questions I was asking about in the while you're waiting assignment. Let's see how we did. So for this first one, question 1, the A's, we've got 33% of you that say A and 66% say B. We've got, let's see, for this tiny little sequence, 36% and 64%. That's interesting. A little pattern emerging. Okay, what do you think is going to happen on the third one? We've got 2% B and 98% C. Oh, I'm sorry. I've done these in the wrong order. It's Monday. Anyway. No, that's right, isn't it? Why did 98% of you pick C? The A. Right, the one difference between these 4 different sequences, there's one change. So what makes that the mutation? What rule are you using in your mind? Most of you, 98% of you answered this correctly. That's the mutant sequence, not A, not B, not D. What's the mental thinking that you're doing in your head that makes you say it's C, it's not A, B, and D?
If you assume that each piece is of the same, it's the only one out of the 4 that's different, so it's easier to assume that it's the mutant.
Yeah, it's like Occam's razor. The simplest explanation is the most likely one, that one of the 4 DNA sequences has a difference. It's majority rules, essentially, in other words. If most of the sequences look identical and there's one difference, that must be the mutant version. So why are the results more mixed than for these top 2? Why didn't we get convergence as a class on one answer for the top 2 examples?
I assume that B had a deletion, but you could assume that A actually had an insertion.
Yeah, so there could be an insertion in this sequence, or there could be a deletion in that sequence.
So the mutation depends on which deletion insertion is.
Right. So this is why, presumably, this is why all of you were split on these questions. What if I had added option C? Now, who would vote for B as being the mutant sequence? The vast majority of the class. In this case, because now at the site that's different, B is the uncommon variety. It's got a T there where the other, or G there where the other 2 sequences have a T. So this is all about the concept that I meant to talk about last class, and I'm only going to talk about really briefly today, which is figuring out the direction of change. That is, what was the original version and which version of a gene might we determine to be the mutant version? So we could start with an example like this. We've got the human chromosome. We've got the chimpanzee chromosome where there were 2 different chromosomes. In humans, those 2 chromosomes make up one whole chromosome like this. So which happened? Did the human chromosome split and chimpanzees have two, or did the two chimpanzee chromosomes fuse and humans have one? What happens if I give you this information, which we've seen before in class? Was this fusion of a chromosome in humans, or was it splitting of chromosome 2 in chimpanzees and in gorillas and in orangutans? Which one's more likely? The fusion. One event somewhere on the branch of evolution leading specifically to us, to our species, to humans, there was a fusion. What's another way to interpret this? Well, you could say, in a different color, that the two chromosomes were fused here, back in time, when there was a common ancestor to all of the primates. Then what would we have to do to make chimpanzees, gorillas, and orangutans all have fissioned chromosomes, split chromosomes, if we started off with the fusion as an ancestral state? If we wanted to have two chromosomes in gorillas, orangutans, and chimpanzees, that means on every one of the lineages leading up to those species, there would have to be fission. So which is more likely? One event, fusion specifically in humans, or fission happening separately, the division of one chromosome into two in three different species? It's way more likely that we had a fusion in the ancestor of all humans. Just a bit of evolutionary thinking to get us going on a Monday morning. But the basic take-home point that I want everybody to appreciate, which most of you do given the Socrative results so far, that you need a lot of evidence to decide whether or not something is a mutation in one individual or on one chromosome or in one species. You have to compare that individual to many others. So this is how biologists determine who is normal or wild type or common. You survey a lot of individuals or a lot of chromosomes or a lot of species, and you ask what's normal, what's typical, what's average. And if you're not that, then you're the mutant. That's why I asked you to watch this movie before last class, which this movie I think makes this point really well. That this is a composite image of currently the world's most typical person. What was it? Han Chinese male, aged 20 to 28 with a cell phone and no bank account and something else. Yeah. But typical is always relative. So what's typical in the entire world? What's typical in this classroom? What's the average person in this classroom, if we define this as our environment? I don't know, maybe we should do some polls or something. Don't leave. I don't know if she's offended by being typical or atypical, I don't know. Right, so we have to be in genetics and in every other language of biology, we have to be always thinking about what we're defining as our population, our study system. Are we talking about everybody in the world? Are we talking about everybody in this classroom? Who is common, who's typical, and who is wild type will vary. I was hoping that, and we'll see if this is true, everybody brought back an answer to this question. In last class, we talked about mutations, and that oxidation of G produces 8-oxo-G. So exposure to reactive oxygen species, free radicals that our mitochondria produce. What happens if this double helix is exposed, so this double helix, this pair of strands of DNA, what happens if it's exposed to reactive oxygen species and then undergoes replication? So what's the first step in replication? You might remember this from previous classes, you might not. The first step is the two strands separate, denature, unwind, unravel, hydrogen bonds holding them together break. So what do we know about 8-oxo-G? Here's the factual knowledge that you need to know. It base pairs with adenine. G is not supposed to base pair with A, but if it's 8-oxo-G, which I'm going to highlight with this asterisk, it will base pair mistakenly with A. Whereas when this chromosome replicates, the rest of the base pairs make their normal hydrogen bonds, make a normal set of base pairs. So you get this new sequence, TA-TA-TA. What about the other strand? Does anything happen to it? Here's the original strand. So what is the other strand in the bottom here? What's its sequence from left to right? Base pairing happens. So T is A pairs with T pairs with A, C. Did anything happen to the C? Exposure to reactive oxygen species is not a G, so nothing happened to the C, so it still base pairs with G, A, T. So we have a mutation, right? There's an A here. For the first time at this position in this chromosome, there's an A that was never there before. So we get two copies after replication, as usual. We get two copies of one chromosome. How many of those look like the original chromosome? How many of those copies? One looks like the original, the bottom one. It's got the same sequence, TAT, CTA, but we've got one mutation. So that's how oxidation induces mutation. So now we have a mutant version of a chromosome. It only affects G's. Yep.
So when it's exposed to the reactive oxygen species, it doesn't change the original DNA double helix right there. It only affects it later during replication.
Right. So immediately, so the question is, what happens to the original molecule? And this is exactly the point I was trying to make. I just haven't made it yet, or very well yet. That this original molecule of DNA that undergoes oxidation to 8-oxo-G, it's still base paired with C. Right? There's still 3 hydrogen bonds, maybe 2 after the mutation, but that double helix, the original double helix that got that chemical change, is still 8-oxo-G and C underneath it. So there's no change in the composition of the original double helix. It's after replication, right? Here, one of the copies that's made from that chromosome has that mutation in it. The G, what would have been a C, has been replaced with an A. So the source of mutation.
Would that same difference in base pair happen during transcription for the mRNA?
So we'll see about the effect in transcription later, but if the mutation changes an A to, or a C to an A, could affect transcription as well. Depends on where on the chromosome that change is happening. So there are a lot of ifs in the response to that question. So we've already talked about determining the directionality of change. In short, again, to summarize, the rule was you look at a lot of information, you figure out what is the majority, and that's what we call normal, common, typical, wild type. Mutant versions are the odd ones out. And today we're talking about 2 different techniques. Right? This chapter is all about techniques. Hopefully everybody saw the video that I uploaded about PCR. So we'll go through a couple of examples, try to apply knowledge about what polymerase chain reaction is and how we use it in genetics. So we start PCR with a single chromosome, shown up here at the top, double-stranded, got the polarity listed. And these sequences with the black underlines are primer sites. So these are parts of a chromosome where we, geneticists, bless you, have decided that this gray area that I've boxed, this part of this chromosome, those 4 nucleotides, those I'm really interested in for some reason. I haven't told you what the reason is yet. But I'm really interested in knowing something about those 4 nucleotides. And I want to make lots and lots and lots and lots of copies of that part of this chromosome. And that's what PCR does, is make lots of copies of a chromosome. So we define the region that we're copying by where we design these primers, the underlying sequences. So what's the first step in a PCR reaction? The videos, the animations that I asked you to watch were really elegant in their use of color. So the first step is to denature, which I'm going to use red, because those videos that you watched, that's an A, right, had the background get redder in the denaturation step. You heat up this chromosome, what happens? The helix splits, just like chromosomal replication, except chromosomal replication doesn't involve heat, but so we denature. So each of these chromosomes becomes single-stranded, with the hydrogen bonds kind of hanging out in the breeze, not paired with anything. So the denaturation is the first step. Then what happens? Step 2, we anneal the primers. So I'm going to use blue both because that's the color I'm using for primers, plus the annealing step happens at a cool temperature. So you have a combination of this chromosome at the top that we started with, 1 double-stranded double helix. We throw in some primers, and at the annealing step, the primers anneal to these molecules. So which primer is going to go here? The first primer on the left or the second primer below it that's on the left? Well, it's going to be complementary to this single-stranded piece of DNA. So we want something that starts at its, remember DNA is anti-parallel, so the 5' end of the other strand will be here. It'll be C, T, A, T, C, with its 3' end there. So that's, that primer anneals there. So the other primer must anneal over here from its 5' end, A, C, following the rules of base pairing, ACTAT, 3'. Yep.
When you're denaturing the DNA, how do you know that the primer sequence you've identified on your molecule will come back together and align in the same way that you saw it?
Most of the time, the DNA sequences for primers are unique enough that you don't have issues with how the chromosomes come back together when they re-anneal. Detailed question. So that's annealing step. So the whatever primer sequences you've added to the mixture, find where they base pair with the template DNA, the chromosome that you added. So we've got primers sitting on the chromosome. What happens in the third step? What's the third step called? Tack. What's tack? You add DNA polymerase, which is the enzyme that does the name of the third step. Boy, DNA polymerase, regardless of what I've written there, sorry. This third step is called extension. This is where DNA synthesis happens. So we have these DNA primers that are base paired with the original template, and they've got free 3' ends. So there's a 5' end, 3' end. So the 3' ends of the 2 primers are pointing towards each other. The primers are going 3' ends in towards the middle. So Taq DNA polymerase, it's often referred to as Taq, T-A-Q. You don't have to know that. Just so in case I accidentally say Taq again, you know what I'm talking about. Taq is a type of DNA polymerase. So after Taq gets added, then it binds to this double-stranded DNA where the primer has base paired with the template and starts actually synthesizing the new second strand of DNA, moving from the 5' end to the 3' end, and it cruises along until, what happens now? It's synthesizing starting at the end of the primer, and it's going towards the 3' end, and then it reaches the end of the chromosome. Now what? Does it keep synthesizing DNA? No. It doesn't know what nucleotides to add because there's no more strand to read from. Right? Taq was reading this strand of DNA and adding in the complementary nucleotides. When it hits the end of the chromosome, there's no more single-stranded DNA to read from, so it just falls off the end. That's it. And the same thing happens in the opposite direction. So Taq DNA polymerase comes and binds to the 3' end of that primer and adds in the appropriate base pairs, G, A, C. That's the end of the chromosome, so synthesis stops. I want you to know that we're going to have time next class to practice some of this. So I'm going quickly today, partly because some of this should be reviewed from the animation videos, but also partly because next class we'll spend some time working on this. So at the end of round 1, we generate, this is the same as what I just drew out on the previous slide. We have 2 double-stranded molecules of DNA, where we started with 1. So at this point, after 1 round, denature, anneal, extend, we have gone from 1 copy of DNA to 2 copies of a double-stranded double helix. What's the difference? How do these pictures differ from what was on the previous slide, the original chromosome?
There's 2 base pairs that are in pair.
There are a couple of base pairs that are missing. And that highlights a very, very important point, that it's where the primers are located, those underlines. It's the outermost ends of those 2 primer sequences that define the part of the chromosome that we make a copy of. So nothing on the chromosome beyond those primers, here in red, will get copied, ultimately. So we decide where we want our primers to be, and that defines how big the PCR product winds up being, what part of the chromosome we copy. So here in round 2, we denature, make those 2 double-stranded double helices denature. So now we have 4 single-stranded template DNA to synthesize from. So once again, the primers will anneal. So this would be the annealing step. We could anneal primers to all of these primer sites that exist. So we've denatured. We anneal the primers to the template DNA, to the copies of the chromosome. What's the next step?
I have a question.
Please.
Does this extend beyond the underlying portion?
So we haven't extended yet, but they will.
But you wrote like the T, like on the very top of the T and the G, and that.
Oh, I'm sorry. Excellent question. I apologize. Yes, the primers only bind to the primer sites. So the primers were 5 nucleotides long. So those are the primers. Thank you for pointing that out. So after the correct primer sequence is anneal to the DNA, what happens next? We extend, right? So after anneal, and extending, here in blue are the primer sequences hybridized to those copies of the chromosome. So let's do a quick poll. Here, we've got one primer that's annealed or base paired with a template molecule. Which end of the primer is right here, this C on the right? Is that 5' or 3' end? It's the 5' end, because DNA always base pairs in an anti-parallel fashion. So the 5' and the 3' ends are next to each other. So which direction for this primer, which direction is Taq DNA polymerase going to synthesize? Towards the left or towards the right? So you're going to go starting at the 3' end and replicate DNA to the left. So we fill in all of the nucleotides based on base pairing rules. And we stop when we hit the end of the chromosome. Now notice, there's a difference here. This is a really, really important difference in this step of PCR, round 2. So if we fill in the extension product of the other primer, how large is the PCR product that results in this round of PCR, round 2? The 2 molecules we just synthesized, how long are they? How many nucleotides? How many?
I count 14.
14. I counted 14. 14 base pairs. What's the distance if we look at how long that product is between the 3' end of one primer site on the top strand and the 3' site of the other primer sequence on the bottom strand? It's 14 nucleotides. So that reinforces this point that where we decide our primers are ultimately produces a piece of DNA that's an exact copy of the genes we started with, the chromosomes we started with. It's exactly as long as we have defined we want it to be. In this case, 14 nucleotides. Oh, okay. So our choice of primers dictates how big the piece of DNA that we generate is going to be. Yeah.
Whenever you do this sort of process, you always need to choose 2 primers, because if you don't, then the Taq will just come in and keep synthesizing the DNA because it doesn't know where to stop.
So the reason for having 2 primers is because if we only had 1 primer, it's a complicated answer. If we only had 1 primer, what would happen? You'd only ever make copies of 1 strand of DNA, not both strands of the original chromosome. So you have to have primers that are complementary to both strands of the chromosome to make double-stranded copies of the chromosome that you're interested in. Okay, so that's, in a very, very, very brief nutshell, how PCR works. Right. So important things to understand for this are the 3 steps: denature, anneal, and extend. What happens during each of those? How the primers bind to DNA, that just like any double-stranded piece of DNA, they bind in an anti-parallel fashion. And that when DNA is replicated, those primers are extended from their 3' end along the chromosome by a DNA polymerase to make the second strand of DNA. So that's all well and good. This is something that you'll practice a lot more in Biology 104 for those of you that are biology majors. You'll actually do PCR reactions in 104 lab. So this is a good start for understanding it when you get there. what is this? What does making copies of DNA help us do in genetics? PCR is a technique, especially those of you that are interested in doing lab research, with those of us that do molecular biology over in Science 1. Most of the labs that I know of do PCR as part of their research. So this is a really, really, really common technique in genetics because it lets us do a lot of things. So you make a bunch of copies of DNA. What does that let us do? Well, it lets us clone genes. So say you've got a gene in one organism, you want to put it in a different organism. You make lots of copies of that gene. It just makes it easier for you to put some of that gene in the organism you're trying to make transgenic, let's say. It's also useful for generating lots of copies of chromosomes for DNA sequencing. So let's say you're a forensic technician and you're out at a crime scene and you find a cigarette butt that somebody's been puffing on. And you want to figure out whose DNA is on that cigarette butt. How many copies of DNA molecules do you think you get on a cigarette butt? I don't know. You tell me. Your guess is as good as mine. Not very many. You get some lip cells, maybe, I suppose, on a cigarette butt. And then you want to take the chromosomes out of those and do something to analyze them. Well, I'll tell you what, you need more than just 100 chromosomes to learn something about a suspect. You need maybe millions of copies of parts of that chromosome so that you can actually go into the laboratory and study that suspect's DNA. So you use PCR or a related technology to make millions, literally, and billions of copies of the chromosome that you found at the crime scene. Or, in this case, we're going to talk quickly about how you can use it to genotype individuals, which is related to forensics. Determining what nucleotides I have or you have at a particular region on a particular chromosome of yours. So I've had some DNA testing. I'm happy to share my results with you. I had a friend that started a genotyping startup company. They send you a cheek swab. You swab the inside of your cheek with a Q-tip. You stick it in a tube. You send it back to them. They extract DNA from your cheek cells. And then I got to order a few different genetic tests. Let's see. I got to ask whether I was predisposed to being an early morning person or a night owl. That was one of the tests they were offering. I got the lactose tolerance test. You can use genotyping to tell if you have lactose persistence or lactose intolerance.
And it's accurate?
It's pretty accurate.
Like, say the night owl thing? Like,
Oh, no, not at all. So, in short answer, there are lots and lots and lots of different regions of different chromosomes that all together probably contribute to our predisposition to being an early morning person or a late night person. And this test just asked at one of those genes, did I have the early morning or the late night version of the gene? So that's not very predictive. Lactose tolerance, though, absolutely. The other one, what did I say? Oh, colorblindness. I already knew I was colorblind, so I was kind of doing a positive control experiment. It turns out I'm colorblind, which I already knew. So these are the sorts of things you can do when you get somebody's DNA and you have the ability to make lots of copies. So that's genotyping. So here is a picture of the analysis of a PCR reaction. So this is an agarose gel. This process is called agarose gel electrophoresis. And so I want to go through how exactly you can use PCR to tell something about inheritance of DNA or your particular genotype. So in this case, we have diagrammed two different chromosomes. So here's chromosome 1 on the top, double-stranded, chromosome 2 on the bottom. So let's pretend like we've got a diploid individual, like me, and the rest of you, presumably. So I've got two copies of this chromosome. The primer sites are underlined in black, like normal. Here's the catch, though. Are these two chromosomes the same? How are they different? Length. So what's the actual molecular difference? What's the DNA sequence difference between the two? So in one of the chromosomes, I have one copy of, let's call it CA. They're in between the primer sites. What about my other chromosome? How many copies? 3. I've got 1, to 3 copies of CAs on my other chromosome. Okay, so chromosome 1 came from, or this top version of the chromosome came, say, from my dad. The bottom one came from my mom. So I have 2 copies of this chromosome. I'm diploid, like you. But one chromosome, the distance between the primers is shorter for the top chromosome. What sort of a product would get produced? What length is that going to be? 5 nucleotides in the first primer sequence, 5 in the second, and 4 in between. So that should produce a DNA product from PCR that's 14 nucleotides. What about the PCR product that gets generated from the chromosome I have that's the one shown here on the bottom? How big will this PCR copy be? 14, the original 14 plus 2 extra copies of 2, so 14 plus 4, or you could count it out, of course, 18 nucleotides. So if you happen to be, like me, heterozygous, term you've probably heard before, you've got differences on your 2 different copies of the same chromosome. If you're heterozygous, you would produce 14 nucleotide and 18 nucleotide PCR products using the same pair of primers. So here's the really important point. Those sequences on both of my chromosomes that contain the primers, shown in blue, are identical. Yes? Same sequences on both copies of my chromosome. So we can throw my chromosomes into this PCR reaction, add the same primers, and the same primers make 2 different versions, 2 different sets of copies of my chromosomes. Copies of the small version, copies of the large version at the same time. So in other words, you could have individuals that have nuclei that have both small or short versions of this chromosome. We've got individuals potentially that have both long versions. So there's a long chromosome and a long chromosome. Or we could have individuals, say crime scenes, suspects, that have one short and one long version. Because we're diploid, we've got 2 copies of each chromosome, so there's only these 3 possibilities. You're either short, short, you're long, long, or you're one of each, short and long, in a diploid. So I've already told you that this individual is called a hetero or different heterozygote. They're heterozygous. What about short, small, and small, or long and long? What term do we use for those? Homozygous. Homo means same.
Dr. Ross?
Yep.
Is when we represent heterozygous using the letters, the capital and the lowercase letters, is that determined by the dominant recessive gene or just by the length of the chromosome?
So we haven't talked about dominance or recessiveness yet, but that doesn't really impact how we annotate heterozygosity. Good question. Okay. So at this point, bottom line, I'm hoping everybody is at least starting to understand the idea that we can use PCR, and this is where we're heading next. We can use PCR to figure out, for anybody's chromosome, anywhere we want to, on any part of any chromosome, we could figure out what size the 2 different versions of their chromosome are. Are they the same? Are the 2 different chromosomes in an individual the same size? Are they homozygous? Or are they 2 different sizes? Are they heterozygous? And we do that using Agarose gel electrophoresis. So this is an example of a gel, which is basically like a piece of Jell-O. So I'm looking at it from the top down, these blue slabs. Think of it as blueberry Jell-O, I guess. This is blue, right? I am colorblind. Yes? Okay. So blueberry Jell-O, looking down on it from the top. We have a series of wells or depressions, holes you could think of in the top of the Jello slab that we could load DNA samples into. So we could add DNA samples. So that's where the DNA goes. That's where it starts. The PCR reaction products. And then we do two things. We apply T, time. So we're looking at this at time equals 0 and later in time. And the other thing we have to do to perform agarose gel electrophoresis, besides having an agarose gel and adding DNA into the wells, we have to apply a, what's V? Voltage. So that's the electrophoresis part. And if you apply electrical current across this gel, don't worry, this is not a physics class, but if we apply an electrical current, then what PCR does is it separates the PCR DNA amplicons based on their size. Because size is the only thing, write it down and underline it, size is the only information we get out of PCR. It's the size of the products that we generated after the PCR reaction was over. That's the only information we get. It's how long the pieces of DNA are. So if what I've drawn up here is accurate, which pieces of DNA go the farthest through the gel in a certain amount of time? It's the small ones, right? The two-letter words have moved the farthest through the gel. And this is true for DNA products as well. Smaller pieces of DNA will move farther through the gel than bigger ones, which stay here on the gel near the wells where we initially added those DNA molecules. Okay. The DNA is negatively charged. So if you apply a current or a voltage, DNA is negatively charged. Which direction would this pair of nucleotides move? To the left or to the right? We've got a positive terminal on the left, we've got a negative electrode on the right. Which way is a negatively charged DNA molecule going to go? Left or right? It's going to go left. Opposites attract. I know. That's as much physics as we're going to do today. The negatively charged DNA is going to move towards a positive potential. So we load DNA. This is a cross-section of a cartoon of a well now. So we load DNA down into a well. So DNA goes in. There's a negative charge at the left. There's a positive charge on the right. So the DNA moves through. You can imagine trying to stuff DNA molecules through Jello. That's basically what's happening. The electrical current is drawing those DNA molecules out of the well towards the positive terminal. And the smaller ones move farther through that really tight matrix of Jello, gelatin. So smaller molecules can move farther, faster. So how many molecules of DNA, how many different sized pieces of DNA are running down lane A? We've got one band here. Those are a bunch, millions of copies of DNA that are exactly that size, all electrophoresing and moving through the gel at the same speed because they're the same size. So all of those copies of DNA stay together. They're evident as a single band on this gel, a band of DNA copies. So we've got one there, second band, third band, fourth band, fifth band. So there were 5 different sizes of DNA that I loaded into well A, and they moved through the gel at different rates. So So I told you, the only information we get out of PCR with gel electrophoresis is relative sizes of pieces of DNA. So now look at well B. What's the largest piece of DNA in well B? Is it on the left side or the right side? Is that the largest piece of DNA that's running from lane B out this direction, or is it the one on the right? Smallest pieces go farther, or larger pieces go farther? Smaller pieces go farther. So we've got small down here, the farthest from the well. So that's the largest band of DNA that's in well B, the circled one. How do we know how big it is? That's the real question. We want to know how big is that piece of DNA? How long is it? How many base pairs? The way we know that is on the same gel, and this is critical for every Agarose gel that's run ever in the world, many, every day. You add a sample of DNA where you know in advance what the sizes of DNA in that mixture are. That's like the positive control, kind of. So it just so happens that we know that this band is 80,000 base pairs long, and this band is 70,000 base pairs long, and that band is 60,000. That's what these numbers here are meant to represent. So it's an internal standard, something we can compare our unknowns to in lanes A, B, and C, and say, okay, so we can figure out now what sizes are these bands. So how big is this band? It's 70, because it's the same, it's gone the exact same distance through the gel as a band that we know is 70,000 base pairs long. Okay. I want to give us a little bit of time